speaker will be Danyelle Townsend from the University of South Carolina, and she's going to talk about redox mediated signaling pathway in liver cancer. Here we go, Maria.
Perfect. Hi, everyone. I'm not an immunologist, so I'm going to switch gears a little bit and talk about redox platforms and how redox platforms can actually be utilized in the drug development and drug discovery process. The one thing that we do all know is that oxidative and nitrosative stress lead to cellular damage that can induce DNA damage, protein damage, as well as mitochondrial dysfunction and cell death. And so it's not surprising that there are highly evolved mechanisms within the cell to overcome this exposure to oxidative and nitrosative stress. And among these, one of the most abundant antioxidants within the cell is glutathione. And glutathione is a tripeptide that's shown in the top panel here, where what we see is that it's a tripeptide of glutamate, cysteine, and glycine. But it's really the cysteine that is the major workhorse within glutathione, because the cysteine has this sulfur thiol within it that is capable of donating this, is capable in its redox state of acting as an antioxidant to donate those unpaired electrons to free radicals. Glutathione exists in two states, in this reduced state, but under oxidizing conditions, this cysteine residue can form a disulfide with another glutathione molecule, forming oxidized glutathione. And it's this ratio of reduced to oxidized that really mandates the redox status within our cells. And we know that the exposure of oxidative and nitrosative stress not only contribute to the disease states, but it also gets dysregulated in cancer as well as other diseases of aging, like neurodegenerative diseases. And this is in part due to the decrease in reduced glutathione levels and an alteration in our capacity, in our antioxidant capacity. Glutathione is typically recognized in its role in metabolism and drug transport, as well as redox homeostasis. But what I'm going to show you today is it's also a cofactor for a post-translational modification called glutathionylation. And this opens up a new opportunity for drug development and drug discovery platforms. So the cysteine that's localized in glutathione is also cysteines that are in our proteins, and cysteines are the least abundant amino acid. And what we do know is that the genome has about 25,000 genes, but through post-translational modifications, you get to over 1 million different opportunities for differential expression and differential responses within the cell. So cysteine residues are targets for post-translational modifications in response to oxidative and nitrosative stress. So they're very, very highly reactive to their local environment. And so these cysteine residues that are in low pKa environment. Not all cysteine residues can be post-translationally modified, but those in low pKa environments are highly reactive. And so this free thiol in the native state for proteins becomes a thiolate anion, and this can combine either with nitric oxide to form nitrosylation, or can form the disulfide in the sulfur here with the cysteine in glutathione to form glutathionylated proteins. And we know a lot about phosphorylation and phosphorylation being regulated by kinases and phosphatases. But what we also know is that glutathionylation, even though it's a less recognized post-translational modification, it's highly regulated by a similar enzymatic cycle. So this is a prototype of a protein that has a free sulfhydryl that under oxidative nitrosative conditions can become this cystenyl radical that leads to glutathionylation. And this occurs through an enzyme called glutathione S transferase. And glutathione S transferase was originally identified as an enzyme that's involved in drug metabolism because it conjugates glutathione to small molecules in the cells to neutralize them and to facilitate export from the cells. And GSTs are highly expressed and overexpressed in the drug-resistant cancer phenotype. And so it was rather interesting to see that GSTs also play a putative role in inducing glutathionylation, which is a bona fide post-translational modification that through glutaredoxin and sulfuredoxin can be reversed, sending the protein back to its native state. So just like phosphorylation, we have glutathionylation, the forward reaction being propagated by GSTs. So GSTs not only play a role in binding to J and K as an inhibitor and altering the apoptotic and proliferative pathways through acting as a negative regulator in those pathways, it mitigates glutathione conjugate formation in drug metabolism, but also glutathionylation of proteins. So about 15 years ago, one of the steps that we took is taking GST-Pi wild type and knockout mice and looking at what are the targets of glutathionylation, and can any of these targets be utilized to identify the cancer phenotype or utilized for drug development purposes. So what we identified were these protein clusters, or we identified hundreds of proteins and then broke them out into clusters, identifying their structure and function. And so what you can see here is that there are a wide variety of proteins that are GST-Pi regulated through glutathionylation that involves signaling, cytoskeletal proteins, as well as transcription factors and energy metabolism. One thing that was rather surprising to us is that there were also a large number of proteins that are involved in protein folding and ER stress. Well, GSTs have been classically noted and recognized as cytosolic proteins. So we did some subcellular localization and found that it's actually highly abundantly expressed in the endoplasmic reticulum. So the ER, just to remind us, is heavily involved in protein folding, calcium homeostasis, and cancer cells have a highly enriched ER because they're highly proliferative cells and overturning a large amount of proteins. So we validated that GST-Pi is a resident ER protein through subcellular distribution studies and utilized GST-Pi wild type and knockout mice to validate that these are in fact ER resident proteins that are being glutathionylated. These are just images showing where we did nail this down as an ER resident protein, but then did proteomics to identify what are the specific targets within the endoplasmic reticulum that are glutathionylated in GST-P wild type versus the knockout mice. And as you can see, there's calnexin, calreticulin, SERCA, and BIP and protein disulfide isomerase. So these indicate that GST-Pi not only regulates our drug-resistant phenotype, but it's doing so in response in orchestrating cellular events within the endoplasmic reticulum that alter calcium mobilization as well as the protein folding process. So we validated that not only are these resident proteins glutathionylated by GSTs, they form these heterodimeric complexes in the process of doing so and facilitate this GST-Pi mediated increase in glutathionylated proteins. But what is the structure and the functionality of some of these targets, and how does this relate to the cancer phenotype, whether it's the resistant phenotype or the sensitive phenotype? We wanted to first look at calcium homeostasis and looking at its role in regulating through modification of SERCA and some of the other calcium mobilization enzyme or proteins. And the reason that this is we had also utilized thapsigargan and tunicamycin, which are two drugs that are used to induce ER stress and alter the protein folding capacity within the cell. But we wanted to compare this directly with, we wanted to compare this directly with Velcade. Velcade is one of the, or bortezomib is utilized for patients with multiple myeloma. And it also induces ER stress. So what we find is that in the GST-Pi competent cells, and here we're looking at bone marrow, since we're looking at bone marrow derived cancers, and the drug-resistant phenotype associated with those. What we found is that when you can glutathionylate proteins, you have a higher capacity for protection or survival mechanisms within those. And so this gives cancer cells an unfortunate, or myeloproliferative cells, that advantage within pro-survival pathways. So we started looking at the drug-resistant phenotype in multiple myeloma and found that multiple myeloma cells that have high levels of GST-Pi expression also have a significantly higher IC50 level to Velcade, meaning that they're far more resistant. So what we did in the next series of studies is take a GST-Pi inhibitor and showed that when you decrease GST-Pi and decrease glutathionylation of ER-resident proteins, you sensitize those cells to Velcade and other ER stress-inducing agents. So this is fairly important because GST-Pi is also reflected in the population through 4 different polymorphisms and different enzyme catalytic activities. And so what you see on this panel here is showing the differential glutathionylation pattern with the different alleles. So this suggests that not only do we have phenotypic differences in our susceptibility to combat the effects of oxidative stress, but also in our ability to develop drug resistance and that drug-resistant phenotype that's seen with Velcade in multiple myeloma patients. So collectively, we developed a model showing that these ER-resident proteins, glutathionylation of these ER-resident proteins can induce pro-survival pathways, and these pro-survival pathways, when shut down, can actually enhance the susceptibility or the sensitivity to ER stress-mediated chemotherapy drugs like Velcade. So it can play a role in drug resistance in multiple myeloma. The other aspect of the ER is the protein folding capacity. And so in addition to calcium mobilization, we have protein folding. Well, with respect to protein folding, we know that ER stress and the abundance of ER stress for a long period of time can lead to something called activation of the unfolded protein response. And so this occurs when our protein folding capacity exceeds the capacity for folding those proteins. And so this is a diagram of that. And it's a little bit complicated, but essentially what's happening is when there are too many proteins or too many proteins to be folded are going into the ER, there is a highly conserved pathway that's called the unfolded protein response. And this is activation of 3 proteins, IRE, PERK, and ATF6 along. And what this does essentially is it leads to translational attenuation or stopping the translation process and decrease so that you can get a handle on folding those proteins that are already in there. As well as inducing transcription factors that can lead to the upregulation of other chaperones to manage the unfolded proteins. And in the event that goes on for too long, you lead to the upregulation of another transcription factor called CHOP, and this leads to ER stress-induced apoptosis. In a different cell model system, we were looking at developing drugs that induce nitric oxide or induce an overwhelming load of ROS and reactive nitrogen species. And this drug is called PABA-NO. So what we found interesting in looking at the mechanism of action of this, we were expecting to see a high level of nitric oxide and a high level of nitrosylated proteins. But what we actually found is a very high level of glutathionylated protein disulfide isomerase. And so we wanted to find out if protein disulfide isomerase, which is opposite to what we had seen in the Velcade story, is associated with this mechanism of cell death. So what we found is that enhanced glutathionylation of PDI is concurrent with activation of all 3 arms of the unfolded protein response. So in other words, it leads to activation of translational attenuation, at regulation of those UPR-induced cell death pathways, and transcriptional activation of those transcription factors that induce chaperones that facilitate protein folding. So what is protein disulfide isomerase? It's not only the most abundant protein within the ER, it's one of the more abundant proteins within the cell in millimolar concentrations. So what it does is three different things. It can induce disulfide bond formation and facilitate protein folding by inducing sulfhydryl disulfide bond formation. When there are misfolded proteins, they can also isomerase or break these disulfide bonds and facilitate the correct folding of those proteins. It also acts as a chaperone, and I'll talk about that towards the end of the talk. So we wanted to know what are the functional consequences of glutathionylation of PDI at the protein level as well as in the cellular level. And so looking through circular dichroism and intrinsic fluorescence, what we identified is there is an alteration both in the secondary as well as the tertiary state of PDI as a consequence of glutathionylation. But what happens to its isomerase and its protein folding capacity? And that's significantly diminished in a dose-dependent manner. So increase in glutathionylation, decrease in the protein folding isomerase capacity. What was interesting, we were already doing subcellular distribution studies, and what we found is that not only is PDI glutathionylated in response to our nitric oxide releasing prodrug, but that also when we were seeing cell death occur, there was also a translocation of PDI from the endoplasmic reticulum to the mitochondrial membrane. And these are colocalization studies, and we use the Pearson coefficient to validate and to sort of quantify this mobilization. So what we found is that the modified PDI, not native PDI, mobilizes to the mitochondria. And so we thought, well, what is it doing there and why is it mobilizing there? So we took isolated mitochondria to do some more specific studies, and what we found is that glutathionylated PDI with isolated intact functional mitochondria lead to cytochrome C release, calcium release, mitochondrial membrane depolarization, as well as caspase activation. So what was surprising to us is that glutathionylated PDI could induce mitochondrial apoptotic markers, but native protein had no effect. You can see the native protein having lesser of an effect on that. So we went and started looking at cellular studies as well as in whole animals looking at this, and have identified that PDI actually does translocate, or its modified form translocates to the mitochondria where it's interacting with proteins and leading to cytochrome C release and calcium activation. Uh-oh. I think it's stuck. So in other studies, we do know that, so in separate studies, I'll kind of give you a little vignette on the side. We've taken GST-pi inhibitors and shown that when you decrease the glutathionylation of PDI, you decrease the mobilization of PDI to the mitochondrial membrane and decrease cellular damage. So this is well and good for things like lung fibrosis and COPD. But in the cancer phenotype, that's not what we want to see. So this is just a cartoon showing the summary of what we found is that following the Pabino, which is a GST-pi activated prodrug utilized in cancer, that we see glutathionylation of PDI, mobilization to the mitochondrial membrane and leading to activation. I had also alluded to the fact that PDI acts as a chaperone. So it wasn't surprising that PDI was kind of jumping around the cell because it's known to be localized also in the cytosol because it interacts directly with estrogen receptor alpha, not estrogen receptor beta. And in doing so, it facilitates its translocation to the nucleus and and stability within the cytosol. So we wanted to kind of take a deeper dive into this, and we moved into a breast cancer model. And this is just showing in estrogen receptor positive breast cancer patients that there's an upregulation of PDI. And what's unfortunate is that upregulation of PDI is also shown in these Kaplan-Meier plots to have a decrease in overall survival in patients. So we wanted to go back to a cell-based model and look at what are the effects of glutathione and the effects of the anti-cancer agent Pavano on ER alpha stability. So what we found is that glutathione of PDI disrupts the protein-protein interaction with ER alpha. So in the cytosol here, you see ER alpha sitting with PDI, HSP70, as well as HSP90. And for functional transcriptional activation, this complex with ER alpha, HSP70, and PDI mobilize and sit on the estrogen receptor element and lead to activation of c-MYC and some other downstream proliferative pathways. But when you glutathionate PDI, you get that disruption and degradation of ER alpha. So it's plausible that targeting PDI and glutathionation of PDI is another alternative way of regulating the estrogen receptor signaling pathway in breast cancer. So this is just showing that when you do disrupt ER alpha with its protein-protein interactions with PDI, you also block its interaction and it therefore has anti-estrogenic signaling aspects within the cell. So looking in both the context of our multiple myeloma studies, our breast cancer studies, and then we also have, I had sort of alluded to the fact that we also work in a lung injury model. What we've identified is that native PDI plays a putative role in protein folding within the ER. But when it gets dysregulated at very high levels for a prolonged period of time, you get mobilization to the mitochondrial membrane where it acts to induce apoptosis through cytochrome C release and caspase activation. But it also leads to dysregulation of estrogenic signaling through its decrease in protein-protein interactions with ER alpha. So what we're currently doing is taking a drug development strategy because, as you notice in the multiple myeloma model, these post-translational modifications, what we've identified with the pro-survival phenotype that, and I didn't have a chance to talk about that today, but the pro-survival phenotype that we identified in the drug-resistant phenotype in multiple myeloma with Velcade is associated with glutathionation of a protein called BIP, and that has a pro-survival capacity. So in this scenario, developing inhibitors of GST-Pi decrease glutathionation and increase the sensitivity to chemotherapeutic drugs, but that's cancer cell dependent. So in the breast cancer model that I showed you, we have an increase in glutathionation that's associated with cell death pathways. So it's the opposite, it's the Janus effect. But this leads to the UPR activation and dysregulation with ER alpha signaling. So we're taking that two-pronged approach in the drug development strategy to mitigate glutathionation patterns both as a pro-survival as well as a pro-apoptotic role where we're developing inhibitors as well as modulators of PDI in other solid cancer tumor models. And on that note, I'd like to thank everyone in my lab as well as my funding sources, which are quite important, particularly these days. So if there are any questions. No? Okay, well, thank you.